nature. Okay. Good afternoon, everyone. Hope you can able to see the questions and also the discussion what I am doing it here. Can you type in the box whether is it visible for you the question paper and also the explanation which I am giving you on the board. Is everything okay? Can you able to see the questions? Hello. Can you able to see the questions? So the second question is which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? So ATP means it's the energy production. For the sake of energy production, which cell organelle among these is mainly responsible? You know that which is the powerhouse of a cell. So the powerhouse of a cell is nothing but it is mitochondria. So the first answer is glycocalyx, and the second one is the mitochondria. That means it is able to produce as it is the powerhouse of the cell, it is able to produce the energy which is present. Yes. Then try to answer the questions, Bhavana. Okay, if everything it is fine. So the third question, all these are the previous year's questions. So, water soluble pigments found in plant cell vacuoles are. So, which are the water soluble pigments? So, here if you can see the options, all the four are the pigments which are responsible. Means if you take chlorophyll, it is mainly responsible for the sake of the photosynthesis and carotenoids, xanthophylls and anthocyanins. All these are the pigments which gives the specific colors. But here they are asking water soluble pigments which are found in the plant cell. So which are the water soluble pigments? Water soluble pigments in the sense these pigments, I'll give you one more clue, they cannot able to cross the plasma membrane. So the third one is anthocyanins. So this anthocyanins are the pigments which cannot able to cross the cell membrane and it won't leak the outer side, and also these are water soluble pigments. For fourth question, I'll give you the time. Which one of the following cell organelles is enclosed by a single membrane? So type the answer for this. Yes, very good. It's visible. Very good. And try to answer the fourth question. I'll give you time for each question, 30 seconds like that. Within that, you have to do it. So that will complete this chapter question. So the fourth one, which one of the following cell organelles is enclosed by a single membrane? Easy question. Yes, that's the answer. Single membrane. I'm waiting. So what are the options they had given here? Let us see the options. So the options are chloroplast. So you know the structure of the chloroplast. So this is the chloroplast structure. There will be two layers, outer membrane and inner membrane. And the next one is lysosomes. So where does the lysosomes are present? If this is a eukaryotic cell, if this is the nucleus, which is a double membrane structure, and endomembrane will be present over here, and then Golgi bodies will be present over here, or Golgi apparatus, and then you can able to see the lysosomes also. So what is the answer? And nuclei I have shown, and mitochondria, you know that it is also double membrane. Very good. Good. Now let us see the next question. Mitochondria and chloroplast, or see the first option and also the second option, and try to find out whether which option is correct. Which one is correct? So, what do you mean by semi-autonomous organelles? What do you mean by semi-autonomous organelles? If you know that, then you can able to answer this. So, read two options and answer the question. Mitochondria and chloroplast. So, your first option, is it correct or not? Semi-autonomous organelles. So, is it correct? I'll give you time for this question. Yes, semi-autonomous organelles. So I will give you a clue whether they are able to self-replicate or not. So mitochondria and chloroplast, will they have the ability to self-replicate or not? Yes. And providing you the answer. So both the mitochondria and chloroplast will contains their own DNA. So own DNA will be present in both chloroplast and also the mitochondria. So in both things there will be its own DNA. So as there is own DNA in both mitochondria and chloroplast, they can able to self-replicate. Replication can occur in both of them. That's the reason they are considered as semi-autonomous organelles. Second option, if you see, they are formed by division of pre-existing organelles and they contain DNA but lack protein synthesizing machinery they had given. So they contain DNA, they have said, but they are not formed from the pre-existing organelles. How they are formed? You know the theory how the mitochondria and chloroplast has been formed, which I have already explained. And they lack protein synthesizing machinery, means they can able to uh, protein synthesis will occur by ribosomes, that is in endoplasmic reticulum. Rough endoplasmic reticulum is mainly responsible for the protein synthesis machinery. So if you see the fifth one, I will check whether you have answered it or not, then I will tell you the answer. I'm typing all the answers. Yes, it is visible, ma'am. But I think it's coming late. It is visible, your answers. You have answered it as B. Let us check whether B is correct or not. So, yes, it is correct. One is true and two is false. Very good. So now, sixth question. Microtubules are the constituents of. So, once you answer, then I will explain. For sixth one, please try to answer. I can able to see your answers. You can type the answer for sixth one. Just type whether it is A or B, C or D. That's it. So the microtubules or the structures where they are present, if you know where the microtubules are present, then it will be very easy to answer this question. Type A, B, or C or D. So where these microtubules are present, okay, I'll give you the time. Sixth answer. microtubules are the constituents of. Okay, the rest of the answers I will be providing you here. The fourth, meanwhile you type the answer for the sixth one. Yay! Very good. So these are the structures which are present in the cilia and also the flagella, centrioles and spindle fibers. These and all, there will be the presence of this. Very good. Now moving on to the seventh question. Cell organelle containing hydrolytic enzyme. Very easy question. Cell organelle containing hydrolytic enzyme. Seventh one. So meanwhile, I will be writing the answers for the other thing. Fourth is B, fifth is B, sixth A, and seventh. What is the answer for seventh question? Cell organelle containing hydrolytic enzyme. Yes, hydrolytic enzyme means its pH also will be more. More acidic pH will be there. Very good. It is the lysosome. Good. Try to answer the next question. Select the wrong statement. Select the wrong statement. So the first statement, if you see, bacterial cell wall is made up of peptidoglycan. So you know how the bacterial cell wall is made up of what? So whether it is correct or not, you have to tell me. And I have given you the picture also in my previous videos. I have completed the chapter. So whoever are watching now, you can check out the lectures of the cell chapter. Here I will be discussing the question. So bacterial cell wall is made up of peptidoglycan and pili and fimbriae are mainly involved in motility of the bacterial cells. So what are pili and fimbriae? So if this is a bacterial cell wall, glycocalyx and plasma membrane, and inside there will be the mesosome structure. So pili and there will be fimbriae will be present. So what is the main function of the pili and fimbriae? Are they involved in motility or what? What is the answer for eighth one? And cyanobacteria lack flagellated cells. Cyanobacteria is nothing but blue-green algae. So they blue-green algae, whether they require the flagella or not, algae it is. And then mycoplasma is a wall-less microorganism. Mycoplasma is nothing but a PP elbow pleuropneumonia-like organism, which is the smallest bacteria or the organism. So what is the answer? Which is the wrong statement? Eighth one. Very good. Very good, ma. B is the wrong statement because pili is mainly responsible for what? Pili for the sake of the conjugation tube formation. This pili is responsible and fimbriae is mainly responsible for what? This fimbriae is mainly responsible for attachment to the rocks or the host. For this attachment to the rocks if they are present near the rocks and to the host. Whereas pili for the sake of conjugation tube. Which is helpful for the locomotion? The cilia which are present, those are helpful for the locomotion. So the answer, the wrong option is B. Very good. Try to answer the ninth one now. Select the mismatch. Which is the mismatch? So the gas vacuoles will be present in green bacteria cells or not. And see the next one, large central vacuoles will be present in animal cells or not. So what is a vacuole? I have clearly explained. If you know what is a vacuole, then it will be easy for you to answer this question. Know the vacuole, where it is present, whether it is present in plant cell or animal cell, large central vacuole, then it is able to answer. And check your answers. Ninth one, seventh B. Seventh is A, ma, not B. Okay. Yeah, ninth is B. Very good. Ninth is B. Now moving on to the 10th question. Which of the following structure is not found in prokaryotic cell? Which one is not found in prokaryotic cell? Easy question again. Nuclear envelope, ribosome, mesosome, plasma. Which is not found. 10th one. Which is not found in a prokaryotic cell? Yes. So nuclear envelope, ribosome, mesosome, and plasma membrane. Which one is not found in prokaryotic cell? You know that prokaryotic cell will contain a nucleoid or otherwise called as incipient nucleus or primitive nucleus will be present. If there is a primitive nucleus or incipient nucleus, then definitely there won't be any nuclear envelope won't be present in a prokaryotic cell. All the other things, ribosome, mesosome, plasma membrane, everything will be present. So let us check whether you have answered it or not. 10th is A. Very good. Next one. I'll share the next one. Yeah. So you have to match the columns and identify the correct option. I'll give you time for this question. So thylakoids and cristae and cisternae and chromatin they had given. So have clearly explained about all these things in my cell chapter lectures. So now it's your time to answer the question. I'll give you 30 seconds of time for this. Easy one. 11th one. Yes. 11th one is B. Very good. I'm proud of you, Ma. You are answering every question right. So next one. So next is Bolbiani rings or the sides of. Actually, about this Bolbiani rings, I have not explained because for NEET 2022 and 2023, you won't get it. Everything is from the NCERT. But for your understanding, but I have explained the name I haven't given for you. It is nothing but what? It is nothing but in a cell, if at all a nucleus will be present, nucleolus also will be present. Inside the nucleolus, whenever the cell is not in a means it is an interface. In the interface, how the chromosomes will be present? They will be present in the form of chromatin-like structures, thread-like structures, which is a chromatin. And all these chromosomes, if they are puffed up together like this at one place, so that is called as Balbiani rings. So those are called as Balbiani rings. So what are Balbiani rings? Balbiani rings are nothing but the chromatins which are present and they are puffed up like this during the interphase whenever the cell is not dividing. So Balbiani rings are the sites of what? Means which synthesis, that it is nothing but we and RNA and protein synthesis. Means for the nucleolus, if you take nucleolus is the main site for the sake of RNA and protein synthesis. This I had already given for you. So the rings which are present over here during the interphase at the time, those are called as Balbiani rings. Please make a note, but mostly you won't get this because it is not there in NCRT. Whatever it is there in NCRT, only that thing you will be getting. But for your understanding sake, I have explained this. So now moving on to the 13th question. I'll give you the answers, that is 12th is D, but 13th question, now try to answer. Hope you have understood what are Balbiani rings. If you want the explanation, type, please explain once again. 13th is B. Okay, very good. Next, 14th one. 14th one, a protoplast is a cell. See the options and try to answer. It is a cell. Protoplast is a cell. See the options: without plasma membrane, or without nucleus, or undergoing division, or without cell wall. So what is a protoplast? I'll check whether you have answered it or not. 14th one, answer the question. Protoplast is a cell. Very good. It is a cell without a cell wall. We have to write 13th one. Oh, 13th we haven't done. 1 second. 13th B, 14th. Yes. Next, 15th one. Yes, 15th. Cellular organelles with membranes or. So, all the cell organelles which have the membranes, that you have to check it and write the answer. Means all the membrane bound organelles. You know about all different cell organelles. So think about them which is having single membrane or double membrane. Here they have asked only the membranes. That's it. They didn't specify whether it is single membrane or double membrane. Just they have asked cellular organelles with membranes. That's it. They didn't specify whether it is single or double membrane. So it's easy to answer. Very good. Super. Next question. And here in 15th one, nuclei, ribosomes and mitochondria. Ribosomes doesn't have any the membranes. So wherever ribosomes is there, that answer you can opt out. And lysosome is having a single membrane, not double membrane. Golgi apparatus also a single membrane, but mitochondria, it is a double membrane structure. Next, 16th question. Solid linear cytoskeletal elements having a diameter of 6 nanometer and made up of a single type of monomer are known as. So I didn't specify the 6 nanometer that and all, but which is the cytoskeleton? I have explained about the cytoskeleton. Based upon that, you can able to answer. 16th one. I don't know. Okay, I will explain you the 16th one. So this is the solid linear cytoskeletal elements. You know about the cytoskeleton. What is cytoskeleton? Cytoskeleton is nothing but the cilia and also the flagella which are present, that will be attached to the, if it is a prokaryotic cell like that, it will be attached to the plasma membrane. Hope you can able to see this. So, or else I'll keep it like this. Yeah, that is attached to the plasma membrane. When it is attached to the plasma membrane, here I have given you the structure of centrioles. How the centrioles will be, and centrioles are mainly responsible for the sake of spindle fiber formation. And this centrioles will contain like this, the structure I had given. So this centrioles will contains the microfilaments which are made up of proteins. So these microfilaments which are made up of proteins are mainly responsible for the movement of the cilia or the flagella. So the answer is microfilaments. So 6 nanometer, if it is considered, it is a microfilament. Whichever you don't know those questions, you maintain a separate book and write the answers there. Question and also the answer. So only this one you didn't answer because I haven't explained about 6 nanometer. So it is the microfilament. So the 16th one is the B. That is nothing but actin filament which is present in the muscle also. So if when you study about the locomotion, then you will be knowing about the actin and myosin filaments. So those are very thin filaments which are present in the cytoskeleton. Okay, now 17th one. This you have to think a little bit and then you can able to answer. Osmotic expansion of a cell kept in water is chiefly regulated by what? You know about osmosis? 17th you have answered already. Very good. It is B. Very good. So the vacuoles which are present, so those are mainly that regulation will be controlled by the vacuoles only. Osmosis means the entry of water by diffusion like that. Hope you can able to see the 18th one. You have to just match the following and you have to find out the correct. They had given centriole, chlorophyll, Christi, and ribozymes. You answered. I'll check your answer whether 18th is A. Very good. Super. Next one. So that means Christi or nothing but what? Christi or centriole which has been given. Centriole. Centriole, it is nothing but with flagella and cilia it will be present. And chlorophyll, it is where it is present, that is in thylakoid, that is grana, it will be present. And Christi, I think that one, Christi is infoldings of the mitochondria and ribozyme is the nucleic acid. Very good. Next one, see the diagram. It is a diagram based question, 19th one. You have which one of the following organelles in the figure correctly matches with its function. So you know all the functions of the cell organelles which are present. So based upon that you can answer. 19th one. I'll check whether you have. Very good. Super. Next 20th one. So the 19th, rough endoplasmic reticulum is mainly responsible for the sake of protein synthesis, whereas smooth endoplasmic reticulum, it is for the sake of lipid synthesis. So if this is the nuclear envelope, in the nuclear envelope, endoplasmic reticulum will be present. If there are ribosomes, then it is called as rough endoplasmic reticulum. If there are no ribosomes, it is smooth endoplasmic reticulum. Smooth endoplasmic reticulum is mainly responsible for the sake of the this one lipid synthesis, whereas rough endoplasmic reticulum for the sake of protein synthesis. And nucleus function, Christy ribosomes everything. 20th one. Have you answered 20th one? What is the 20th? Major site for synthesis of lipids. Just now we have discussed. So definitely you might have answered 20B, 21D. Yes. 20th and 21st. The Golgi complex plays a major role in post-transcriptional modifications of proteins and glycosylation of lipids. Very good. 22nd. So ribosomal RNA is actively synthesized in. Easy question this also. Where it is actively synthesized? It is synthesized in the nucleolus. Very good. Very good. I will be sharing the next page. Yes. So here it's difficult for you to see, what is true about the. I'll check the book and I'll let you know the question. What is true about ribosomes? They are asking about the ribosomes. So you know about the ribosomes. Prokaryotes will have 70S and eukaryotes will have 80S. I'm not sure, ma'am. I think it's the 23rd one. No, check once again. The answer is wrong. So check once again. So what is D they had given? These are self-splicing introns of some RNAs. No, what do you mean by splicing? In second year you will get this. What is splicing? Removal of introns is called as splicing. In a DNA or a gene there will be both exons and introns. This you will be studying in molecular basis of inheritance. So that removal of introns is called as splicing. So ribosomes are not the self-splicing introns of some RNAs. So ribosomes are mainly responsible for the protein synthesis. So it will contain both ribonucleic acid and proteins. So that is the answer. Ribonucleic acid and proteins will be present. Okay. Next, 24th one. Have the questions in, okay. In Telegram you can see the questions. Okay, good. 24th is B. So select the correct options, correct statement from the following. Proteins make up to 60 to 70% of the cell membrane. Proteins will make 60 to 70, not only in RBC, not in the cell. They are asking about the cell membrane. So cell membrane, how much percentage of proteins will be present? In fluid mosaic model I had given for the RBC, red blood cells. See the other options. Who has proposed the fluid mosaic model? Fluid mosaic model. 24th is B. Yes, good. Next one, 25th. Very good, Ma. You have studied almost all the questions you have answered. The 24th, I thought question was incorrect. Okay, but you are answering all the questions. Good. Next one. Which one of the following organisms is not an example of eukaryotic cell? Easy question. You know what is a prokaryotic cell, pro and eukaryotic cell? Yes, 25th. Easy question. Prokaryotic cell means it will contains all the bacteria. So, waiting for the answer. We don't know. 25th. So, Escherichia coli. It is a bacteria. Okay, whereas euglena, this and all, if I do the biological classification, then you can easily identify the answer for this. So Escherichia coli is a bacteria, and rest of them which has been given, euglena, amoeba, and paramecium, they will come under the protozoans. Okay, write it down. Whichever answers you haven't answered, please write it down in separate book. So rest of them are protozoans. They are not bacteria. Okay. Bacteria is Escherichia coli. In biological classification, definitely you will be knowing about this. 25th is A. So next one, 26th one. Important site for formation of glycoproteins and glycolipids. So glycoproteins and glycolipids, where it will be formed? Easy question. Have you answered it? I'll check it. Yes, very good. Golgi apparatus. Golgi apparatus, 26th one. Next question. Which one of the following also acts as a catalyst in a bacterial cell? This you don't know because when we study about the biological classification and also the molecular basis of inheritance, the next chapter which I am going to deal with, because it is the third most important weightage chapter. In that, you will be understanding the this question, which will act as a catalyst, that and all. So here I'll give you the answer. It is 23rd is rRNA. So if you take the bacterial cell, it is made up of 70S ribosomes. So one larger subunit and one smaller subunit will be there. So this together it is 70S ribosomes will be present. So in the 70S ribosomes also the larger subunit and the smaller subunit will be there. There will be 23S, 5S. In molecular basis, I will be explaining clearly. 5.8 S rRNA will be there. So in among that 23 S rRNA is mainly responsible for the sake of the, means it will act like a catalyst. It increases the speed of the reaction. So S is nothing but here S is the Swedberg unit, or otherwise called as sedimentation coefficient. So no need to worry about this. In molecular basis of inheritance, you will clearly know this. But for now, you can write it down this. Yes, it is nothing but Swedberg unit, or it is sedimentation coefficient. So it acts like a catalyst. So the 27th answer is C. 28th you can answer. So I will be erasing this. Now answer 28th one. 28th is C. 27th also C, I think. Your guess is correct, but in NEET examination, please don't guess it like that, okay? Because you will lose 1 mark. If you are thorough, then only try to answer the question. 28th is C, correct? Good. And next 29th. Which one of the following has its own DNA? Easy question. Hope you might have answered already. 29th one. Own DNA. Which one it has? Yes, very good. Mitochondria. Good. Next, 30th question. The main arena of various types of activities of a cell. Easy question, this also. Means how you can say whether cell is living or it's non-living. How you can say whether cell is living or non-living. 30th A. Various types of activities of a cell, where it will occur. Plasma membrane or cytoplasm. When you consider the cell as a living, because if this is a cell, nucleus will be there. In the cytoplasm, there will be the all cell organelles. These cell organelles are mainly responsible for the formation of everything. Why you have thought it is plasma membrane means I know that in fluid mosaic model we have understood that there will be lipid bilayer, and because of the movement of the lipids a little bit, rarely the movement of lipids, and the proteins will be there. That's why you might have thought plasma membrane, but the organelles which are present are mainly responsible for the sake of the uh, cell to be alive. For that, so it is the cytoplasm, not plasma membrane. Okay, because in that there will be several cell organelles. Okay, got confused with the option, but okay, good. 30 is the C. Next one. Which one of the following structures between two adjacent cells is an effective transport pathway? So see the options and try to answer it. Here I will explain this question, 31st question, because it's you have to know what is plasmodesmata, what is plasma lemma, that and all you have to know. And endoplasmic reticulum, you know the answer. And plastoquinones, those are nothing but the in electron transfer chain, ETC, in the plant physiology, you will be studying about the plastoquinones. So, plastoquinones are nothing but these are the units which are used in the electron transport chain. Okay? That we will be studying in the plant physiology. In ETC, it will be mainly used. And plasma lemma is nothing but the outer covering of the plasma membrane is called as plasma lemma. So plasma lemma means if this is a plasma membrane, the layer which is present on the plasma membrane is called as plasma lemma. And plasmodesmata means if this is one cell and if this is another cell, the gap between one cell to another cell means interaction between the cells, how it will occur through a connection which is called as plasmodesmata. So this plasmodesmata is mainly responsible for the sake of the connection between the two cells and between any two cells. So that is nothing but openings or pores will be present, and through this openings or pores there will be the connection. So the answer is A for 31st one. And what is a plastoquinone? In plant physiology we will be studying, and that is it will be it is one of the component in electron transport chain, and plasma lemma, the outer covering, whereas plasmodesmata, these are the pores or the openings which are present on the cells, so that the two means the cells which are in between can be connected, and then the transfer will happen. 31st explanation I had given, and now 32nd one you have already answered. You have to listen which you don't know also. 32nd is B. I'll check what's the question. The plasma membrane consists mainly of proteins embedded in phospholipid bilayer. Very good. That is fluid mosaic model you have been studied. Please make note of these things. And next 32nd one, you have already answered it. So B, good, very good. Next 33rd one. Cytoskeleton is made up of. Easy question again. 33rd is also easy question. Cytoskeleton is made up of. 33rd D, proteinaceous filaments. Very good. Next 34th. What is 34th question? There is no DNA in. Easy question again. DNA, where it is present, if you know that. Yeah. Okay, good, ma. Next, easy question, 34th. I'm happy that you have studied the cell chapter, have all answered all the questions. Like this, you have to continue for all the chapters also. Then definitely you will get a neat. Okay. So 34th one. 34th, what's the question? There is no DNA in which there is no nucleus, then there won't be any DNA. Mature RBC. Yes, very good. Next one. Middle lamella is mainly composed of. So what is middle lamella? Do you know this or shall I explain this? Okay, I will tell you what is middle lamella, then you can able to tell me. So middle lamella is nothing but the material. You can able to see this slide. The material between intercellular material that is called as middle lamella. So the material which is present in between the cells is called as middle lamella. Now you can able to answer. It is mainly composed of what? Calcium peptide. Very good. Super. So not only calcium, if it is chlorophyll like that, there will be the magnesium peptides also. Calcium and magnesium peptides. Good. And you know what is hemicellulose, what is muramic acid, and also phosphoglycerides. Phosphoglycerides we will be studying in the next chapter in detail also. Next, very good, na? Super. 36th is plasmodesmata or today itself we have studied and it is there. So what's the answer? D. Connections between the adjacent cells. Very good. So I'll be erasing this. 36th is D. And now we will from 37th question. 37th you don't know. Keeping in view the fluid mosaic model. Let us see the question. Keeping in view the fluid mosaic model for the structure of cell membrane, which one of the following statements is correct with respect to the movement of lipids, proteins from one lipid monolayer to the other? Flip-flop movement. For this, you know the answer, but you are not applying it to that. So I have already told you in cell membrane, plasma membrane when we have discussed, it is made up of lipid bilayer. Saturated hydrocarbons and unsaturated hydrocarbons will be present. And in between, there will be the proteins. Some proteins are embedded in the cell membrane and some proteins will be present outside. So these type of proteins will be present and the lipid bilayer will be present like this. Okay. So the lipids which are present, they will move. But the proteins will not move. That too, proteins won't move. They will be present. They will act like a transport. For the sake of transport, they will be useful. The proteins do not move, whereas lipids will move. That too, it won't move very fast. Slowly, like a mosaic movement will be there. So, rarely, slowly, the movement will be there by the lipids, whereas proteins won't be there. And that movement will give you the quasi-nature to the cell membrane or plasma membrane. Quasi-fluid nature will be given. So, now you can answer the question. See the options and answer the question now. 37th one. 37th, now you understood, no? You can able to answer. 38th, you doesn't know. Okay, that you will be knowing in the plant physiology. So, have you understood the answer for 37th? So, lipids can rarely flip-flop, but the proteins cannot. That means it is a B. Rarely they will flip-flop, but proteins never ever flip-flop. So, this is the 37th answer. And 38th one, the two subunits of ribosome remain united. So, larger subunit and the smaller subunit, how they will be united and forms a ribosome, that they are asking. This is different and this is different. So, how they will form into a ribosome, the smaller and bigger subunit. So, you know that ribosome means it is a protein synthesis. For the sake of any synthesis of protein, if it is required. So, you can't able to see the ribosome thing here. I will draw it here. So, the larger subunit will be there and the smaller subunit will be there. These two subunits, whenever they join, then it is called as ribosome. Ribosome is mainly responsible for the sake of protein synthesis. So from this, there will be the protein synthesis. Protein synthesis in the sense food formation, like that it is required. So chlorophyll is mainly responsible for synthesis of the food in the plants. So chlorophyll will contain which ion, or which it contains what? It will contain the magnesium. If magnesium is present, then only it can able to synthesize the chlorophyll. Okay. So magnesium is present. That's it. The answer is magnesium. And here, for the chlorophyll, iron is also responsible to synthesize the magnesium. But here, iron option is not there. So, magnesium will be the correct answer. For the formation of magnesium, iron is also required in some amount. Okay. So, the 38th option is C. Hope you are taking notes of this. 38th is C. 37th is D. Next, 39th question. Okay, I have to share, I think. Okay, 1 second. Yeah, can you able to see the questions now? 39th is D. What is 39th? Vacuole. In a plant cell, is a membrane bound and contains water and excretory substances. Very good. Next, 40th. So, in germinating seeds, fatty acids are degraded exclusively in the. So, the degradation of fatty acids, where it will occur, that they are asking. So, have you typed the answer? If not, I will explain that one. 39B, 40B, Glyoxysomes, very good. So you know what are glyoxysomes? Glyoxysomes are nothing but they are the cells of the yeast which are present in the yeast. Whereas peroxisomes, what is the main function? Peroxisomes are present in all photosynthetic cells. They will be present in the photosynthetic cells. And mitochondria, you know that it is a site of aerobic respiration. Very good. And 41st, wrong statement you have to select. What is the wrong statement in that? Chloroplasts and mitochondria contain an inner and outer membrane. Okay, B. So chloroplasts and mitochondria have an internal compartment, thylakoid. Yes. So here thylakoid will be present only in the chloroplast, not in the mitochondria. Very good. Next. Which of the following statements regarding mitochondrial membrane is not correct? You should know about the membrane of mitochondria. There will be outer membrane and inner membrane. 41st is B factor. So 42nd, if you know the structure of mitochondria, then it will be easy to answer the 42nd question. Yes, not correct. You have to see it is not correct. 42nd A. Enzymes of the electron transfer chain are embedded in the outer membrane. Very good. So if this is a mitochondria, inner membrane will be present. So on the inner membrane, electron transfer chain will be present, not on the outer membrane. Very good. Next, 43rd one. So for this, you should have an idea about the practicals and the microscopes. So I have already explained that way all the inventions and new inventions can be done, has been done after the invention of the electron microscope. So what is present in the electron microscope, how it is helpful to us. So if you know about what is resolution and what is magnification, then it is easy for you to answer. Very good. So the resolution power here of the electron microscope is much more higher than the light microscope. For that reason, it will be taken. Good. Very good. 44. Main organelle involved in modification and routing of newly synthesized proteins to their destinations is endoplasmic reticulum. Very good, ma. Superb. Next question. According to widely accepted fluid mosaic model, cell membranes or semi-fluids where lipids or integral proteins can lipids and integral proteins can diffuse randomly. In recent years, this model has been modified in several respects. In this regard, which of the following statement is incorrect? They're asking about the incorrect statement regarding fluid mosaic model. A. Proteins can travel within the lipid bilayer. Today only I have explained about this. Proteins cannot undergo flip-flop movement. Lipids, they will undergo rarely flip-flop movement. So which is the wrong option? Check once again. 45th. Protein. 45th. Please check it. 45th is the wrong answer you have given. Proteins cannot flip-flop. Only lipids will flip-flop. So 45th answer is D. Check this. Okay, first you check 45th answer is B, and 46th protein synthesis in an animal cell occurs. Ma'am, what is cytosol? Cytosol is nothing but the in the cytoplasm the presence of the extra matrix which is present other than the cell organelles. That is the cytosol. Okay, matrix which is present other than the cell organelles is the cytosol. Okay, then now we can answer 46. You answered it as D. No, check the 46th answer also. It is D. These two answers, please check it. Okay, you typed wrong option. Okay. 46, protein synthesis in an animal cell occurs. Where the protein synthesis occurs? In the rough endoplasmic reticulum and also in the cytoplasm. Okay. Rough endoplasmic reticulum are mainly responsible for the sake of protein synthesis, and the cytoplasm. In the cytoplasm also ribosomes will be present. Okay. So I will stop here for today. So rest of the questions I have already shared to you in the Telegram app. So what you have to do, from each chapter, I will be taking one class for discussing the questions. The rest of the questions, at least you try to complete it today. And tomorrow, what I will do, I'll do the families, three families or leftover, no? That families I have explained to you. If you have doubt, I'll continue, or else we'll solve the morphology of flowering plants questions as we have completed it. So let me know whether you have doubt in the families or not. So do you have doubt in the families? Means that floral diagram. 46 is B. 46 only on ribosomes attached to nuclear envelope and endoplasmic reticulum. No, cytoplasm also there will be the ribosomes. So 46 is D. Okay. So please solve the rest of the questions and send me the answers for that. Okay. And if you have any doubts related to the families, let me know, or else tomorrow we will be discussing about the morphology of the flowering plants, previous year's NEET questions. Type it whether you want the discussion of the floral diagrams of three families, or let us solve the questions, previous year NEET questions of morphology of flowering plants. I think you are thorough with the families, so the differences between the families. I'll send you the notes of that. Okay. Is it okay? Today I will be sending you the notes of that in Telegram. Take that notes, and if you find any difficulty, then I will discuss again. But be thorough with the morphology of flowering plants tomorrow, so that we will be discussing the questions from the previous year. Is it okay, ma? Yes. Yes. So be thorough with the morphology of the flowering plants. notes also I will send to you through the Telegram. Okay, notes along with that. Okay then, bye.